Hello and welcome to Science Research Weekly, Season 4, Episode 26, Massive Spinning Phenomena in Physics. Get ready for Parkinson's disease sniffing dogs, the bladed microtube scheme for massive magnetic fields, a giant spinning black hole merger, ghost galaxies, lasagna plots in SAS, and an NSF grant on Programmable Cloud Laboratories. This episode covers material from July 11th to July 17th. Let's get started with our research articles. First up is trained dogs can detect the odor of Parkinson's disease. Two dogs were trained to detect Parkinson's disease by smell. The researchers used a double-blind trial of 100 samples, and the two dogs ended up having a sensitivity of 70 and 80%, so that's the correct ID of Parkinson's patients, and a specificity of 90 to 98%, that is, correctly ignoring control patients. This is really helpful because there's really no definitive diagnostic test for Parkinson's, biomarker or otherwise, that has been developed so far. So if dogs can do it, or dogs can even help us understand accurate biomarkers, that's going to be a huge step on detecting Parkinson's disease so we can better treat it, better look at etiology, etc. All right, next article is Gigagauss magnetic field generation by bladed microtube implosion. Researchers used a bladed microtube target with a slanted sawtooth-like pattern that was highly irradiated by laser pulses, and this produced a rapid plasma implosion and generated ultra-high magnetic fields. They call the setup the bladed microtube, or BMI scheme, and it represents a new way to create high magnetic fields in a very compact space. Let's switch over to cell biology. There's the article, Dynamic Organization of Cells in Colonic Epithelium is encoded by 5 biological rules. So via tissue renewal modeling, researchers established what they would consider 5 mathematical laws as a set of rules for at least colonic epithelia, that type of cell. The laws were 1, the timing of cell division, 2, temporal order of cell division, 3, spatial direction of cell division, 4, number of cell divisions, and 5, cell lifespans. And those rules can sort of form the basis of organization of cells, especially during, I think specifically during continuous crypt renewal. And they might more broadly help us understand the mechanisms that underlie how cells organize into tissue types and how disorganization, how dysbiosis may lead to things like cancer. All right, I think the biggest sort of news of the week was the paper from the preprint, GW231123, a binary black hole merger with total mass 190 to 265 solar masses. So biggest news in astronomy, I went right to the source, this archive preprint. So a gravitational wave signal from the merger of 2 large black holes was detected, and they were both spinning very rapidly. And actually, they're spinning so fast that they're actually near the limit of how fast they could even spin based on Einstein's theory of general relativity. So it's like almost violating what we know about the laws of physics. And then also the size of these black holes, these are really big black holes, too large to have been from sort of a standard stellar collapse, so suggesting an intermediate mass black hole. So not like a supermassive black hole at the center of a galaxy, not just a stellar mass black hole, but larger ones that had already formed larger through maybe other mergers. So very interesting, sort of on the edge of what we know about physics, cosmological phenomena, and kind of rewriting a little bit of what we think those limits are. Okay, switching over to this next article, Encoding Hierarchical 3D Architectures through Inverse Design of Programmable Mons. So it's pretty complicated details, but I think the main point's fairly straightforward, at least for me, or at least for my interpretation of it. The researchers were essentially fabricating nanoscale material using DNA. With that, let's turn to our final one here. 100 ghost galaxies may be orbiting the Milky Way, and we're just now uncovering them. This final article is just a news article, no journal paper yet. But researchers using supercomputing methods, they suggested there are actually 80 to 100 satellite galaxies orbiting our galaxy, orbiting the Milky Way, and they were too faint to really be seen before due to them being stripped of their dark matter. So we couldn't look at, I guess, gravitational interactions. And that dark matter is possibly stripped by the halo of our galaxy, assuming that all works out with dark matter. So, I mean, I think another term they use in this article is orphan galaxies, that they're kind of small, faint, kind of on their own, and it was hard to detect until now. Okay, let's switch over to some research tools. First of all, the General Statistical Software, volume 113 is now available with several articles already up. There's SCPI and certainty quantification for synthetic control methods. There's Survey HLM, a SAS macro for multi-level analysis with large-scale educational assessment data. StepMix, a Python package for pseudo-likelihood estimation of generalized mixture models with external variables. Bayes Mortality Plus, a package in R for Bayesian mortality modeling. And STOP, with 2 P's, an R package for spatial temporal point pattern analysis. Over on the SAS front, Rick Wicklund from The Do Loop has this new article, Use a Lasagna Plot to Visualize US Gas Prices. And it's how to use a lasagna plot to visualize, again, the gas prices as a simpler and more interpretable method than a horizon plot. He saw this kind of large, if you're watching the video, you can see it, sort of a large, hard to interpret, kind of clunky horizon plot, and he used SAS code, and actually created a much more interpretable one, especially with color shading. You can really see the gas prices across time. Okay, over on R, 3 new R bloggers post. First, from Bioconductor Community Blog, there's an announcement that there's a online developer forum on July 28th. It's Exploring R and Rust in Bioinformatics. Next, from Nicola Rennie, How to Choose an IDE. That's a integrated development environment. It's not really an endorsement it's not an endorsement of a specific IDE, but more of questions to ask when deciding which one to choose. And then finally, there is from a Statistician's R notebook, standard deviation versus standard error, meaning misuse and the math behind the confusion. Always good to have a refresher on fundamental concepts. There were 6 new CRAN packages I thought you might want to know about. There's RFriend, provides batch functions and visualization for basic statistical procedures. There's Cluster Web App, universal clustering analysis platform. Fused Tree, fused partition regression for clinical and omics data. Iron Seed, improve random number generator seeding. Junko, create common tables and listings using in clinical trials. And ggTranslate, a ggplot extension for translating plot text. Finally, 2 grants of note. First, from NSF, National Science Foundation, the testbed towards a network of programmable cloud laboratories. The program seeks to establish and facilitate the operation of distributed autonomous laboratory facilities where the idea of programmable cloud libraries, they're able to essentially execute automated work streams, including self-driving lab workflows to move research goals to AI, and enable experimental design, laboratory preparation, data collection, data analysis, and interpretation, sort of in a more, again, can be distributed, can be more remote, can be more standardized, because if like you have one system doing everything, you can tweak it and shift the code, but then when you have the same code, it should run the same thing and be as close to things as possible. Whereas you could have the same protocol, but different, say, experimenters running it slightly differently. So some of that reproducibility can be diminished. And of course, access. Like if you don't have high-tech bioinformatics platforms, it's hard to do those things. But if you have a cloud lab that you can pay and submit a job to, That's a lot easier than finding capital to build or buy your own. All right, enough of my waxing poetic about cloud laboratories. Let's go to our final grant, the NASA Roses 2025. Sort of omnibus is open. There's a lot of different programs. I selected one I thought of note is the F9. It's the Citizen Science Seed Funding Program. It's aims to support scientists and other experts to develop citizen science projects and to expand the pool of professional scientists who use citizen science techniques in their science investigations. Gigagauss in the house. Thanks for joining me for Science Research Weekly. Science on.